Hi everyone, I'm Sudeep Tiwari. Thank you very much for giving me this opportunity to present my work. The topic of my presentation is direct foliar uptake of phosphorus from desert dust. But before going into detail about the talk, first of all, I want to ask one question. How many of us will believe that this desert dust can be a good source for the nutrients? I think none, or even maybe very few people. But after finishing my talk, you will be able to perceive this idea that desert dust can be a good source of nutrients to the plants. So it was Dr. Avner Gross who conceived this idea that a desert dust can be a good source of nutrients to the plants in nutrient deficient conditions. So to prove this, that desert dust can be a source for the nutrients, he collaborated with Dr. Ranheral in Gilat Research Center, and they conducted the initial experiments to see that desert dust can be beneficial to the plants or not. And that was a eureka moment for them. They saw that yes, desert dust can be a good source for the nutrients. And for further carrying the research, they they undertook the studies on different model plants. So their hypothesis was the desert dust can be a good source for the nutrients, and they have few questions to answer to prove this hypothesis. So the questions spans around dust capture on the leaf surface and retention, dust p-solubilization, p-transport through the leaf cuticle. So these are the three important components which must be answered to prove this theory that dust can be a good source for the nutrients to the plants. So since we cannot take all the nutrients at a time, so we concentrated on the phosphorus. This is one of the most important nutrients for the plants for the growth and development, and one of the limiting nutrients also. So we conducted series of experiments and in under the glasshouse conditions with three model plants, that is chickpea, wheat, and maize. So chickpea and wheat, it originated to this dusty region, and maize, it is originated far from the dust source that is near to the Central America. So we have here we have two different nutrient or irrigation system, that is with the P sufficient and P devoid. So there was no phosphorus supplied. So when we found there was a visible symptoms of the P starved plants, we started dusting on them, and the dust was done in the pulses, in the three pulses. So after dusting, and we waited for 2 weeks, and then we terminated the experiments recording all the parameters. And it was surprising to us that we found that the P star plants, they were able to utilize the dust as a source of phosphorus, and they have doubled their biomass than comparison to the minus P plants. This was also similar in the case of the wheat, but we found there was a marginal difference in the maize plants. So to see the different questions, we first looked that what are the basically reasons behind the dust holding or dust retention capacity, as they have to retain, and then they have to process it so that they can utilize for their growth and development. So we conducted experiment to see the dust holding capacity and dust retention capacity. So in the dust holding capacity, we found that the minus P plants, they were able to hold more dust than comparison to the plus P, that is the P sufficient plants. And it was similar in the case of wheat, but maize was less responsive. In case of dust retention capacity also, at the end of the experiment, we saw that chickpea, in the minus P plants, they were retaining higher dust than comparison to the plus P plants, and in the maize the response was very less. So from here we can see that under the starvation, the plants can capture more dust and retain it for longer time so that they can solubilize. Further, we looked into the structural and chemical modifications. So in the structural modifications, we see that under the starvation there is structural modifications like trichome density. The minus p plants, they have the higher trichome density, they have the higher exudation, and they have higher stomata than comparison to the plus p plants. We also see that there is the trichomes, they are the points of entry for the solubilized p. Further look into the chemical changes that occurs when there is a pea starvation. So we found that there is a change in the leaf pH in the chickpea, wheat, and maize. We found that in chickpea the pH was much lower than comparison to wheat and maize, and there was a higher solubilization in chickpea than comparison to wheat and maize. Further, we looked for the organic acids like oxalic acid and malic acid. They are the most important organic acid in the surface metabolites that we have mined to the literature, and we found that these organic acids are secreted much higher in in the minus P in the P star plants than comparison to the plus P plants. To prove this, we conducted some in vitro experiments, and in the in vitro experiments, like with the different pH and with the different organic acids, and we found that the the hypothesis was true, and the plants they lower down their pH, and then the solubilization occurs more, and p is released. Similar is with the case of the organic acids. When there is a secretion of higher organic acids, they can solubilize more, and there will be more release of phosphorus. Further, we looked into the metabolite profile, basically the surface metabolites that we have collected, and we found that there is a huge difference between the starved and non-starved plants. However, we were interested only with the oxalic acids, and we didn't look to the other metabolites and their role in the dust solubilization. But we have found that the oxalic acid, malic acid, and the other organic acids may be responsible for the release of the phosphorus, which plants utilize for their growth and development. Further, we want to see that what happens to the dust when that is deposited on the leaves. So we we did analysis, and we found that the post dusting there was a change in the dust chemistry, and it was surprising to us that the maximum portion of the unavailable P was utilized by the plants. So to prove this hypothesis, we conducted an experiment with the commercially available phosphorus, that is apatite, that is calcium phosphate, that is in general not available to the plants. So when we dusted in the similar way as we dusted the desert dust to the plants, we found that the results are almost similar. They doubled their biomass than comparison to the minus P plants. So it was proved that plants can utilize the unavailable P pools and for their growth and development. Further, we want to see that what happens to the P sufficient plants. So we also supplied the dust to the P sufficient plants, and we found that there was a marginal difference in chickpea plants. However, there was no response in the maize plants. Further, we also looked that what will happen when we have a foliar fertilizer applied to the plants, to all the different plants, and we found the similar pattern of results that were found in the dust when it was applied. Similarly, in the P sufficient plants, we also find the similar kind of results. There was no response to the fertilizer. So in the summary of this study, we see that in the first of all I will start with the figure B. So from this we can see that in general, theoretically, it is considered as the nutrients, basically the phosphorus that is absorbed by the roots, and there is exudation process and all everything. But the thing is that the plants, they have to compete with other microorganisms, they have to compete with other species, and they are also soft to the other minerals. So that is almost limited for the plants for the growth and development. But when they are supplied from the atmosphere, we can see that there is a less competition and the plants have adaptive mechanism to solubilize this and use for their growth and development. They are also highly enriched than comparison to the what is available in the soil. So concluding our study, we say that chickpea and wheat, which originated basically in the dusty regions, they have adaptations to utilize dust as a source of pea. They have adaptive modifications that leads them to have high holding dust capacity and retention capacity. Dust, dust can also uh, have a modifications, that is structural and chemical modifications. They have decrease in the pH, they have increase in the exudation and the organic acids, so that helps in the solubilization of the phosphorus, and that can be utilized by the plants. Further, we also found that dust p acquisitions were not shown in the maize plants, which is originated in far away from the dusty region. And we also found that there was no response to the MKP in the P sufficient plants. Further questions that we are going to ask is the characterization of microbiome, how the microbiome, phylosphere microbiome, basically they are going to help in P solubilization under the P limited conditions. We are also going to see what are the conditions which will maximize the P solubilization, like wettability and organic acid, microbe secondary metabolites. Further, we have also interest in to look the other sources like biomass burning, and we also want to look for the sources like iron, zinc and potassium, which can be supplied from the dust. If you are interested to go more in this study, you can see the link below for our publication. You can go through it. Thank you very much for your kind attention. Namaste.